Thanks a lot for this nice introduction, and to ChemAxon to give me the opportunity to share with you some of the current activity we have in AstraZeneca to increase our compound collection value and diversity, and this through collaboration, partnership, and open innovation. So I would like to start by to apologize to two persons. One is Metse, because I will add this conference to the long list of conferences where people will hear about what I will say next slide. So the challenge in drug discovery. So he wanted to avoid that, but no. And the second is the movie maker, that I'm not good in movie, because I was filmed already in a conference 2 years ago, and after 2 years, I just have 70 views in YouTube. So this doesn't match Lady Gaga, in fact. And but I got one like, but just for my wife. So so I invite you to go there. So so most of you knows about the challenge we are facing in drug discovery. So in one hand, the number of drugs approved with respect to the R&D investment decreased with the time. This picture is taken from an article where the author showed that many series of drugs are in fact targeting a small set of biological targets, sometimes all targets. And of course, there is this trend to reduce the internal investment, so the investment in internal R&D capabilities. So a lot of persons in the industry agree that we need to increase the collaboration, the academic group, and also between pharma companies. So AstraZeneca set up a series of strategy around that, around early discovery partnership, and these are the remit, is to first to find a new and validated target to treat disease, and to access through collaboration, new chemistry space, something that we don't have in our compound collection, and also we cannot access using our chemist expertise, and also to deliver lead optimization project in a new way. So this is a busy slide, but just on the top panel here, you can see that the collaborations we put in place to try to increase the value of our screening collection. So in fact, finding new targets for our existing compounds. So for example, we regularly screen a subset of our collection, 250,000 compounds, to screening center like LDC Pharma, MRCT, and Neomet that suggest a new innovative target. Importantly, if AstraZeneca is no more interested in one of the targets, this screening center, this group, having also chemists, can have the option to pursue the project for themselves with the AstraZeneca compound. So we are also part of this AMI Lead Factory Consortium, where a series of pharma companies share around 50,000 compounds of their collection to be merged in a huge, not so huge, but a screening collection that can be screened by the other partner, but also by academic group and CRO that don't have maybe access to this large set of compounds. And we also screen our compound on target related to neglected disease in collaboration with DNDi. So on the bottom panel is the collaboration we are currently working on to increase the diversity of our compound collection and the diversity of the chemical space we screen in our project. So we continuously design and synthesize a new chemical library in collaboration with Pharmaron in China. We screen XChem, so DNA encoded library with XChem. And my talk will be more about this panel here, especially these two collaboration with Bayer and this open innovation. And that's one of the main reasons I'm invited here, because it's related to ChemAxon. So in fact, there are 3 parts in my talk. The first will be about this HTS screening collaboration we have with Bayer. The second is to show you how we gather compounds from academic and how do we profile them through our open innovation portal. And then also I take the opportunity to give you an overview about what we usually consider when we analyze external collection. So the first part is the collaboration with Bayer. It's called Boomerang. It started in 2010, and it was extended until 2016 so far, as it's working well. So in fact, within this collaboration, the two companies nominate a target, an HTS target, and to be screened by the collection of the other company. So to nominate a target, of course, the target should not be of common interest, and should not be also in a research area that are where the two companies work on. So it's quite restrictive, but when the target is nominated, they exchange all the information to be able to run the assay and also follow-up assay, not only the HTS, but also the secondary screening and so on. So, if this is successful, and it was several times, not always, that we found interesting series, and then we can exchange chemical series between the two companies, and these series are reviewed by dedicated chemists. So at the time, we compare the two collections to understand about how much the two collections overlap. We used 2D fingerprint, and we found that only 3% of these two collections overlap. And most of these 3% are, in fact, due to the fact that the two companies make component acquisition to supplier. And so in fact, there is no real overlap between the two collections or proprietary compounds. So this means that when you nominate a target, you can be screened to more than 4.2 million unique structure. And today we are more close to 5, in fact, because this is all result. So this is just show you one way we can use to largely increase the chemical space we screen on a target. And it's a relevant chemical space in first approximation because it's Bayer, it's a pharma company, so a competitor. So the compounds already have some more sense than just random compound, I would say. And second part, so about gathering compound from academic groups of small CRO. So this can be done through this open innovation portal that is quite new in AstraZeneca. We are not the first one to suggest this kind of portal. And especially on this link here, when an academic group can use to submit compound to all things. So how this work? So the partner, so the academic group or small CRO can submit novel compound, we hope, to be part of the AstraZeneca HTS collection for 1 year. So we make first a quick assessment if the compounds are novel, and also some limit, but quite relaxed limits on the FISChem. If the compounds succeed this criteria, I would say, AstraZeneca is just support the transfer of the sample, the real material, to the HTS center in England. So, and then of course, we are engaged to report any activity we found on the compound. And what is really important is also that the academic group retain the full intellectual property of their compound. They own the compound. We can just discuss options about what we can do with the compound if they are really interesting after a certain screen. And so when we decide to set up this environment, we have few requirements. We don't want, before we get the sample, to see any structure. So and this person said that because there is a risk of IP, intellectual property contamination. So, but in other way, that as a compound is supposed to come from many universities around the world, we need something, tools to standardize the structure in order to make a proper novelty check to our collection. So there are different ways to do that, and the way we choose is to outsource this task to a third party, and over the few companies that we screened for this task, ChemAxon was selected as the preferred partner. So their work is to establish legal agreement with the submitter and to manage the structure transfer, to their server, where they can then standardize correctly the structure, generate the fingerprint, and then run the novelty check against our collection. And also PubChem as a proxy to see if the compound already exists in the public domain. And also they identify if there are controlled substance, can be an issue if the compound comes from outside England. And also they run some property calculation, and they compile all this into a report that is sent to us for review. So here is just to mention that we use Scholar 1 from Thomson Reuters to use used for the file transfer between the different entities. And also here that we update every quarter the collection that ChemAxon used for the novelty check, just to get a novelty check as up to date as possible. So this is just a snapshot of the ChemAxon Cheminformatics report we get. So it's a little bit longer, but it's really what we need. Some profile about the molecular properties that give us just a flavor about how the compound looks like. Is there no molecular weight more than 1,000 or things like that, or slope P more than 10. But most importantly, what we look directly is this similarity to our collection. So if there is a peak at 1, meaning that the compound or around 1 is really, really similar, one of the compounds is really similar to one of our compounds, we have the option to remove this compound from the submission, and then we are clean with this IP contamination. And also some structural filter, quite useful to see if the submitter, the academic, would put metal or isotope in their compound. That's not what we are firstly interested in. So we agreed with ChemAxon to extend, to enrich this Cheminformatics report, not just because we want to filter out the compounds that the academic group submit, because we are quite open and we just focus on the novelty and not really on the properties. But we found that it could be really interesting for this partner to get a profile, a more deep profile of their compounds. So not just getting there to have their compound screen, but also getting some computational data on their compound that can be interesting to maybe engage discussion. So we agreed to implement the result that we can be computed using this interface. It's called BioClips. It was developed by AstraZeneca and University of Uppsala. It contains AstraZeneca QSAR model to predict solubility, log-D. And we will have to talk about how to update this, because this is based on auto-QSAR. It's a method we have that every time that there is a new measurement, a log-D measurement, the model every week is updated, is retrained, and the previous model, the new model is checked with the previous one and see if it's a useful new model. So we see. So I'll go to the third part, and then I will show you some what we usually look when we receive an external collection, or we have to analyze external collection. So first things to novelty for AstraZeneca, and that to check this we use a platform that we have in house called Chemistry Connect. It's just a repository of a list of chemical databases, public or commercial databases. Altogether, it's 86 million records. So this is really used. Of course, the AstraZeneca collection is there. So this is used to see if the compound is in AstraZeneca, but is the compound already mostly in PubChem? Are you really interested by this collection or not? So, and also it's a nice way to always have hands on what kind of new compounds comes out in literature or in patents. The second thing is that we always compute this compound filter. Again, it's not to filter, it's more to tag the compound. And for that we have our AZID filter, which contains two different kind of filter. The property filter, you can see, is really quite relaxed Lipinski rule. So if a compound violates one of these properties, called a backup, is the compound just match one of the unwanted substructure or reactive function, it's called an ugly. And then if a compound does not violate any of these criteria, it's a core compound. And here it's just the profile for the AstraZeneca collection. It's just most of our collection is core compounds. In fact, the idea behind that is just the core compound can enter the HTS collection. You need specific requests if you want to screen other type of compounds. And we have also a bunch of other kind of substructure. Next one is FISChem. You can imagine that molecular weight CLOP are mandatory. But I will not list all the FISChem property we compute on an external collection. Just give you an example here. What we found often in literature is the profile number of aromatic rings or aliphatic rings. But we found recently more useful to combine this information into one here. So I don't think you can read this, but in fact it's just the number of aromatic underscore number of aliphatic. So this is just an example. If you see this peak there at 20%, meaning that the collection from partner 1 has 20 compounds based on 2 aromatic, 1 aliphatic. So, of course, can be an infinity of possibilities, but sometimes we have a profile where another collection can have 40% and or specific pattern. So, it's at the end, it's more informative that to use these two separated. And about molecular complexity, I think that's really one of the most important descriptor we have to analyze today, I would say, for us. It's, of course, the popular fraction of sp3 and the principal moment of inertia, like here. You see, as you know, a compound according this value fall within this triangle, when on this corner you have the compound that's more rod, and on this one you have more spherical compound, and here more disc compounds. So, and the person who published this say that they are putting, so this is just a kind of a way, a proxy to get an idea of the shape of the compound. And people show that they are putting binding pockets that likes to see compound here in the middle or more in the right of this triangle, where most of the corporate collections and tendency to sit on the left side of these triangles. So it's quite approximative and quite useful, especially when we decide to in-license a library from Alchemia, which contain this very particular sugar scaffold. Of course, the bulk of the discussion was about, do we need this kind of scaffold within our collection? But having this plot here was really facilitated the discussion, since you see here for the AstraZeneca collection, we have really on the left side, and we can really directly with a simple plot really demonstrate that we can visit other part of the chemical space by acquiring these libraries. Another one about molecular complexity, because again it's really important. This is, I will not explain this in details. It's just based on the work of Peter Earle from Novartis. He just showed how useful it can be to build a Bayesian model to discriminate between what looks more synthetic compound and what looks more natural product compounds. So we have reproduced this model using our data. And so we have this kind of score. The more you are on the left, you have more aromatic, more synthetic compounds. The more on the right, the more your compounds start to look more like a natural product. So you see here when you analyze the collection from partner in red here, we could not see their structure. It was not alone. But using this plot, we really see that their distribution within this plot is skewed on the right. So and if you use this limit, you say, okay, this collection might enrich our collection to something that looks more natural product. And you see AstraZeneca is not as good as we want today. So again, it's very easy to compute, really things useful to analyze. We also start to increase the number of substructure we do. So we are like many other big pharma, looking for macrocycle and other kind of really specific 2D structure. But also we tend also to run more and more virtual screening using 3D shape and especially the ones that are related to alpha helix mimetics and beta strand, and also protein-protein interaction. So we have a series of query that comes directly from the literature, and we build more and more query using our internal data. So this is run in batch against a new collection, external collection, and we see if they are interested to acquire this collection or to in-license to have more such compound in your collection. Of course, what we had also is everything based on 2D fingerprint that is really common and really powerful. So in two ways, in fact, we like to analyze how similar the external compounds are close to or similar to active compound from Campbell or from GVK Bio database, especially the one that our collection is not similar to. So we want them to have a kind of biological annotation of this new compound to tag if this one is close to an active compound for which we have nothing. And also the diversity. We will give preference to compounds that are relatively diverse to our collection. And also the same technique is used for clustering. When it comes to the selection, it's clear that we want to cover as maximum the collection, depending on how many compounds we can select, to cover a maximum of cluster of this external collection, also to reduce singleton exposure. When I will say that we are not that obsessed to exclude any singletons, we are not that afraid from them. And also last but not least, when we have the opportunity, we like to have an opinion on a medicinal case on the top of our data, just to say, okay, this scaffold talk to me, or it say nothing, or this compound is not stable. So all these things are presented in the third part. Is of course run on Linux cluster. So also we have a portable version that is built on Pipe and Pilot. That is, we have case where the company could not send the structure, and we wanted to get some profile, and then we just compile a protocol that we send to them, and just say run the protocol and just send us and keep it if you are interested. But just send back the PDF report, and then we can see how it looks like. Then we can engage further discussion if you found this profile interesting. So you see, in this case, we don't have any IP issue. We have, of course, no limitation of CPU calculation. And also, we are limited by the choice of this platform. But maybe Michel is here and thinking, why not implementing in KNIME? I would say, yes, we can. So, and also we build this infrastructure that is able to run on a single laptop. You know that computer has become more and more powerful. But for example, all the things that I show you in the last few slides can run on a single laptop. And for example, just don't have exact benchmark, but structure standardization to start with, then novelty calculation, molecular complexity descriptor, Fischem properties, Mursko scaffold. I didn't talk about that, but you probably know what it is. All the filter. It just takes 1 hour to run for 1 million compounds. So we don't see any need to increase the performance, because when we need this isolated laptop with no network and ready to format after, is because we meet someone with a very sensible collection. And like another pharma, for example, this means also that what's the size? 2-3 million? So I think it's perfectly fine in the morning to get already all this profile. So we don't need. So for the 2D fingerprint, it's still challenging. We are 12 hours for 2 millions, again 2 millions. It's still okay, because when we meet, we just like to spend time together. So it's okay. We don't need a few minutes for that. But I think, I don't know what's all this match with ChemAxon tools, but I heard yesterday 1 million, again 1 million takes 30 minutes. So I don't know for 1 million here. Maybe that range also. But I don't bet on this. I tried to log in from my home to check and to give you the answer, but I could not. Okay, thank you. And the 3D shape, of course, is still challenging, but usually when we use that is because we apply already a a part of the funnel, and we do this on a smaller set. But any improve in speed of running 3D shape similarity really useful. And conclusion. So my talk, I describe the HTS partnership we have with Bayer as a very good way to increase the chemical space around the screen target. And I described the AstraZeneca Open Innovation Portal, especially the one to gather compounds from academic groups, and also give you an overview about what we usually look, what property we use to make a decision, or to have just an idea about how an external collection can be helpful for us. And then there is some acknowledgement related to different parts of the talk. Thank you very much. Thank you.